So the recording's in progress. Ollie, if you want to go ahead and kick us off with your presentation, it is all yours. Thank you.
Great. So yeah, thanks for the invite. I mean, we've been following Dosh for a while, so it's great to be involved and to speak. So I'm a researcher, so I'm a marine biologist. I spend lots of my time on the beach. But when most people go to the beach, they're interested in the big things, so the crabs, the fish, the jellyfish. Whereas I am interested in the things that everyone else walks over or on. So interested in baby or embryonic marine animals. Being a marine biologist interested in early life stages is a bit like being a sort of child in a sweet shop, because there's just a huge variety to choose from. So everything from a sort of snail with two hearts that beat with completely different rhythms. So beat with completely different rhythms and are then reabsorbed during the process of embryonic development, through to jellyfish that are having to swim to stay alive, because as soon as they sink and touch the seafloor, they stick to the seafloor and die. And then here on the right, we've got jellyfish polyps. So these are sort of small blobs of jelly which effectively metamorphose and become sort of the jellyfish that everyone knows and loves. So we've got a huge diversity of forms when we compare different species. And even within a species, within a week, these animals can look very, very different. PowerPoint is not playing. So this video is showing sort of 5 days difference, and you've got animals that are sort of spinning around, essentially like a football inside an egg. So that's the center video here. Idea of scale, these animals are 1 millimeter in length. So the embryo is a millimeter in length. That's its kind of maximum size. And we go from this sort of ball of cells spinning around inside an egg through to a fully formed snail down here on the left-hand side. And that's all happening dynamically out in the environment. And what we're particularly interested in at the moment is this concept of the Anthropocene. So humans are having the biggest impact we've ever had on the world, and that's obviously impacting animals in all sorts of different ways. And one of the things that's been quite slow to kind of catch up, we're talking about how we can try to sort of mitigate our effect, but how we're actually measuring our effect on the animals that are living out in the environment has really sort of lagged quite far behind. And one of the key things here is that early life stages, so baby marine animals, if you like, are more sensitive to what goes on around them in those later life stages, because they're having to put themselves together at the same time as sort of survive the environment. So what we're about is the idea of using imaging approaches to go from video, so video in the sort of top left-hand corner, using computer vision to identify animals, to track them through time, analyze how they're behaving, how they're performing in all sorts of different ways. So we measure their heart rate, we look at fluctuations in pixel values, and we generate huge volumes of data. And we've been doing this for about 15 years, and we started using microscopes. So we started, with microscopes on the bench top, and very quickly you start to realize the limitations of that kind of an approach, because somebody would come and sort of lean on the bench and you'd end up with lots of vibrations, or you wouldn't have fine control over the environment, which would cause problems to your experiments. And we went to all of the sort of the major microscope manufacturers, and the technology that we wanted didn't exist. So what we've done is built a sort of series of open source technologies using approaches like 3D printing, lots of like low cost electronics. And the aim here is to create an instrument, create a capacity that we can then make a sort of a global standard. And the reason why that's particularly important is because we need to be using standardized approaches to assess sensitivity of animals to climate change and to pollutants and to all sorts of different environmental stresses. So the key here was not just to sort of develop an instrument that would do a job, but I think like lots of open source projects, it became more than that, which was, okay, well, what parts can you reliably get hold of? How can you make this something that people could actually kind of create with limited technical information? And yeah, how could we make it scalable? So we've sort of taken the I guess the ideas here from kind of 3D printing and 3D printer technology. So we're kind of using stepper motors and belts to move things around. So we're moving a camera underneath a multi-well plate. So a multi-well plate is something scientists use for culturing animals. So essentially what it allows us to do is to look at individual animals using a sort of robotic camera, and to repeat that process all the way through that animal's development. So if the animal develops over a week, then we capture video maybe every hour over that whole week-long period. And we can then combine all of that together to see what's going on, see what that animal did. And we work on lots of different animals, lots of different species, but for some species, this is particularly a serious problem. So Antarctic species develop over sort of months, or in some cases even years. And without like these kind of robotic or autonomous approaches, it's just an impossible task. So this is a paper that's currently in review. So we're trying to get this published in Hardware X. And we've got a build guide, which we could put online. And we have tried to make it as accessible as possible. So we've embedded sort of CAD models. I'm hoping you can see the browser, but we've sort of embedded CAD models into the build guide itself that you can kind of manipulate. You can kind of go in and look at individual parts. So it's trying to get the information out there in a form that enables somebody to build this from the ground up, but also go in and redesign it and make it bigger and make it better. And we're using both open source hardware, but we're also really heavily using open source software here. So everything we create, we make open source. We're also drawing on other on packages, other projects. And the other instrument that I just want to touch on more briefly is essentially a kind of a comparable technology for actually putting out in the environment. So this lab EmbryoCam is designed to be in a lab, but actually it could be in a school, a college, it could be in a in a field station. This next technology is designed to be out in the environment itself, attached to the sea floor. And the idea here is that you're actually imaging, so you're actually sort of capturing video in a similar way, animals out in the environment. So we can actually see what they're doing. We can see how they're responding. I mean, all over the world, we're seeing more extreme events, and particularly here, we're sort of seeing animals exposed to temperatures that our lab experiments suggest they shouldn't be able to tolerate. So rather than try and work that out in the lab, we're developing these technologies that allow us to sort of see that firsthand. And the combination of these two then allows us to kind of twin or join up the laboratory and the field, and actually start to sort of take data from one to use as a way of kind of informing the other. And we're working with sort of international collaborators here. So the aim, I mean, we're using this for our own research, but it's got really broad applicability. So in particular, we're working with kind of the companies who are responsible for making sure only safe levels of chemicals go into the environment. But we're also working with organizations involved in things like aquaculture. So could you use these technologies to make aquaculture better, more sustainable, and sort of less harmful for the environment? Okay, so I've rattled through that at pace, and so I think I'm open to questions, and I'm more than happy to, given the pace, to go back and revisit any of what I've described.